While studying the male reproductive system, coming back to the male reproductive system, I told you it consists of a pair of testes. This is the primary sex organ. We had done all about the testis and the scrotum. Accessory glands was left, duct system was left, and the copulatory organ was left. So under the duct system, you have epididymis, which is not, you have the epididymis, you have the vas deferens, and glands like prostrate. Then you have the seminal vesicle. Then you have the Cowper's glands also. So there are three glands, three accessory glands, and epididymis, vas deferens, all of these form the duct system. So today's class, we are going to do the duct system and the glands. Now look at this. This is the structure of epididymis. This is the structure of epididymis. Epididymis, as in you can see, this is the testis. And from the testis, now see, inside the testis, as I told you, the tunica albuginea divides the testis into compartments. And each testis has testicular lobules. They has lobules and seminiferous tubules. All right, and seminiferous tubules then form these straight tubules, which merge and form this. What is this structure? This netted kind of structure. This is the rete testis. This is the rete testis, and here is again a structure which is the vasa efferentia. And from here, this green structure that is shown over here is the epididymis. So this epididymis, you have the head of the epididymis, which is called the caput. You have the body of the epididymis, and you have the tail of the epididymis, that is cauda. So what does the epididymis do? What role does the epididymis have? That is very important. You do get questions based on epididymis. So first of all, testis plus epididymis is known as testicles. In the common language, we often refer to as testicles. So testis plus epididymis called testicle. If you get an MCQs, you should be able to do. Each epididymis is around, it's a long tube, 7 to 12 meters, highly coiled. That's why it fits into this area. It's highly coiled. And what does it do? Why do we require, why the male reproductive system? Why does the male reproductive system require the epididymis? So this is 7 to 12 meters coil, and it is this site for temporary storage of sperms. Sperms are temporarily, so it is the temporary storage of sperms. It is also the site for maturity. Sperms, see, sperms which are released, or spermatozoa which are released from the testis, no, they are not active. All right. So what happens to them? They are not active. So as these sperms travel from the seminiferous tubules to the rete testis via the vasa efferentia to the epididymis, they are activated by the fluids present inside the epididymis and the vas deferens. So basically they are inactive at this point of time. If you just take out sperms and in vitro and try to fuse it with ovum, they will not fuse because they have not attained that activeness. So this epididymis is a place where the sperms are temporarily stored and they attain maturity and motility also, the ability to swim. They are inactive, not so active. They are just moved on. Okay, and there, while they are inside the epididymis, their nature changes. They become more and more motile, more active. So the pattern of motility and the percentage of motility, that means the ability to swim, the ability to move, changes as the sperms travel from the head of the epididymis to the head to the tail of the epididymis. Now, for what amount of time do they remain inside the epididymis? That is a question. So they remain inside the epididymis up to around, a month or so. And once they are ejaculated, that means once they are out of the, but the sperms are not active. So in order for the sperms to be active, they travel in the epididymis, which is the temporary storage. Then they travel to the vas deferens, which is the secondary storage. Then they meet the secretions of the various glands. All those secretions get added, and when they are ejaculated out in the female reproductive tract, they developed the ability to swim. So all the time, how do they move? Now you should ask me that, ma'am, how are they moving if they are not active? So basically, all the time inside the testis and in the epididymis and everywhere, they move, they are propelled by the fluid movements, the fluid secreted by the ejaculatory duct and the vas deferens, and the peristaltic movement in the vas deferens basically helps in the movement of the sperms. You're understanding? But totally they become active when they reach the female reproductive tract. And over there's a process called, where they are activated, and they receive secretions, and they travel to the ovida. Over here, you will see that the various glands like the seminal vesicle, it adds up various nutritive substances into the semen like fructose. So we will discuss about all that. So what we have done for epididymis is around 7 to 12 meters long. It's coiled, and testis plus epididymis is called testicle. It is the site for temporary storage, maturity, and motility of sperms. Sperms remain in the, they are stored in the epididymis. But see, what happens is sperm production is a continuous process. All the time ejaculation is not taking place. So what is happening to the sperms? So this epididymis is also a place, also acts like a recycling unit. The phagocytic cells present in the epididymis, they digest the sperms. Otherwise, always there will be an overflow. Understanding? So this is also a function of the epididymis, is to it acts like a recycling unit. It acts like a recycling unit. So it digests the sperms. It digests the sperms all the time. The sperms are being produced. So the recycling center of the sperms is the epididymis. So all the time phagocytosis takes place inside the epididymis. So understood. So what is the function of the epididymis? The function of the epididymis is to temporarily store the sperms, provide maturity, the ability to move slowly the sperms become active. Also, it helps in phagocytosing the sperms. Okay, so this is the epididymis, the first duct. They can remain in the epididymis for around a month. So they can stay in the epididymis for around a month. And something you should know, that once ejaculated, once the sperm is out of the male's body and is into the female reproductive tract, the total time till which it can survive is approximately 3 days, 72 hours. But the fertilization should take place within 48 hours. Otherwise, the sperms will die. So it is active once being ejaculated into the female reproductive tract. It is active for around 3 days, around 72 hours. But yes, 48 hours, like 2 days. I mean, after 2 days, they mostly die. And during those days, when they have reached this the female reproductive tract after such a long journey, um, less amount of sperm survive, and because there is difference in the pH of the male and female reproductive tract. So when we do the process of fertilization, I'm going to tell you what are the hindrances and how much effort the sperm makes to travel such a long distance through the female reproductive tract, and what are the barriers it has to overcome. Understanding. So this is about the epididymis. So if they ask you a question, so questions asked over here is which is the temporary storage, which is the recycling unit for sperms. So your answer will be epididymis. Your answer will be epididymis. Now the second accessory structure or the duct is your vas deferens. Vas deferens. Vas deferens, you can see the vas deferens is over here, and vas deferens is around 45 centimeters. So it's like small, not so long like the epididymis. So it is around 45 centimeters in length, and it ascends along the posterior body of the epididymis. So see where the location is. Look at this. So look at this diagram. You can see this is the head of the epididymis. This is the body, and this is the tail, the cauda part. So from here, from the cauda part arises this structure is the vas deferens, okay, or the sperm duct, which you call. So the sperm duct arises from the posterior body of the epididymis. So this is the posterior part. So this is arising, this vas deferens is arising from the posterior most part. And where does it go? It goes into the abdomen, right? So look at this diagram, it will be clear to you. So these are the testes, and this is the epididymis. You can see the posterior border. So this is covering the vas deferens's location is posterior border of the, touches the posterior border of the epididymis. And then I told you about spermatic cord, right? This is the spermatic cord. Spermatic cord is the cord through which the testis remains suspended. The scrotal sacs are suspended through the spermatic cord, and this is the inguinal canal, the passage through which it comes out. The scrotum comes out, right? So it takes the same route. The vas deferens take the same route. So they ascend along. They ascend. That means going up. Descend means coming down. Ascend means going up. So the vas deferens ascends along the posterior body of the epididymis through the spermatic cord, and then enters the pelvic cavity. So it is entering the pelvic cavity. Now inside the pelvic cavity, it makes a loop over the ureter. It makes a loop over the ureter. You have the ureter, all right? So it makes a loop over the ureter and then passes over side and down the posterior surface of the urinary bladder. Just a second. Let's look at this diagram. Where is this is the vas deferens. Can you see? So it is making a loop over the ureter. It is making a loop over the ureter and passes side and down through the posterior surface. And then, over here, it broadens to form a region called ampulla. So this is also a place where sperms are stored. So this is the site for secondary storage. And then as it moves down, it meets the duct of the seminal vesicle. And these two together, where the duct of the seminal vesicle meets the sperm duct or the vas deferens, it is called the ejaculatory duct. Understood? So what all we have mentioned about, or which is significant about the vas deferens. The vas deferens, 45 centimeters in length, loops over the ureter and passes to the posterior surface of the urinary bladder. Okay, and the dilated portion of the vas deferens or the ductus deferens is ampulla. So they ask you about this ampulla. Ampulla is also present here. This is the secondary storehouse of the sperms. So which is the primary storehouse and which is the secondary storehouse is important. All right. So this is the secondary storehouse of the sperm. Look at this diagram carefully. What are they showing? They're showing testes, then they're showing epididymis, then see how the vas deferens is arising and moving into the pelvic cavity, and then it will loop around the urinary bladder. That I had shown in the next diagram. Can you see this portion? From here, a small portion of the vas deferens has been cut and these ends are ligated. So this is the surgical method of birth control, which is called vasectomy. So once the vasectomy is done, so the sperms are produced, but they do not, the semen is produced, but it does not contain sperms. But this technique is not active, like, just you've done and, it's sperms are not going to be there. It takes some amount of time so that, because the sperms are present here also. So all these sperms should get exhausted. So after being made, they are stored. I told you this is the secondary storehouse. So sperms are being stored. So till all the sperms over here are exhausted, it cannot be a birth control measure. So you have to wait after a vasectomy done. If a couple does not want children, then they have to take care of this, that all the sperms in the upper portion of the vas deferens should be exhausted. All right. So understood, epididymis and vas deferens. If they are not ejaculated to the female, so they are being they die, and there's this process of phagocytosis which is taking place in the epididymis also, and also in the some amount is taking place in the vas deferens, but primarily in the epididymis. I told you, the epididymis is the primary recycling unit. I mean, where due to phagocytosis, the sperms are digested also. So all the time, no, they are digested. That means the phagocytic cells will come and engulf them. Now look at this diagram. This diagram shows accessory struct glands in the male, and there are 3 important glands. You can see a pair of seminal vesicles, and then you can see ejaculatory duct where the sperm duct meets the seminal vesicle. This is a gland called the prostrate gland, and then 2 other glands which are in a pair. This is the bulbourethral, one on each side, or the corpus gland. So you can see that this is the urinary bladder, and into the urinary bladder you have this these ducts opening. So this is the urinogenital passage in case of male. So in male, there is the passage is common for urine and sperms, but at a time either urine or sperms can come out. So in the coming up classes, I will explain you the different regions of this urethra. So urethra is divided into 3 regions. There is this prostrate urethra near the prostrate gland. Then there is membranous urethra, and then there is this lower part, penile urethra, that enters the penis or the penis. All right. So this we are going to do in the coming up classes. Now coming to the glands. What are the different glands? You have the seminal vesicle. They are in a pair. They are very important. 60 to 70% of the secretion. the semen is made by the seminal vesicle. Then you have the prostate, that is only one, that is also a very important gland, and the bulbourethral or Copper's gland. What does the seminal vesicle produce? The seminal vesicle produces the seminal fluid. Seminal fluid it makes up around 60 to 70%. Seminal vesicle secretes a viscous fluid that accounts or contributes to 60% of the total volume of the semen. Semen is actually sperms plus the secretions of all the glands. And what does it contain? It contains three important substances, very important substances, without which the sperm cannot be active. Fructose, that provides energy to the sperms. Fructose, that provides energy to the sperms. Fibrinogen, that helps it to clot. And a very important substance called prostaglandins. This is very important. What are prostaglandins? They are like derivatives of arachidonic acid. And what is the role of this prostaglandins? That after ejaculation, when the sperms reach the female reproductive tract, these prostaglandins, they induce mild contractions in the uterus, so that there is this upward movement of sperms. That means sperms are forced upward towards the oviduct. You understanding? So prostaglandins are very, very important. So in the next class, we're going to continue from here, where I'm going to tell you about the accessory glands, and then we will study about penis and the different parts of the penis, and then I will explain you the process of. We will do a comparative account of the male and female secondary sexual organs and the homology between them. That is a very important question for NEET. And then we will start the process of fertilization. Okay.